May I now take the pleasure of introducing our speaker for today's webinar, Dr. Ajith Kamath. Well, Dr. Ajith Kamath has 25 plus years of preclinical drug discovery experience, primarily at Pfizer Worldwide R&D USA. He obtained his Ph.D. in Biochemistry from the Indian Institute of Science and carried out postdoctoral studies at the University of Texas. Dr. Ajith joined Pfizer's Research and Development Center in Groton and Cambridge and worked there for 13 plus years as an associate director. After 20 years in the US, he returned to India and served as a senior VP at Jubilant Biosystems for 4 years. Then he rejoined Pfizer as the head of external R&D innovation, worldwide R&D, Pfizer India, and successfully initiated collaborations with Indian pharma companies and academia. His team contributed to 2 drugs, one in the market and one is in the clinical trial. Dr. Ajith also worked at Mitra Biotech as executive director to lead their external research efforts. And currently, he is an advisor to 2 startup companies, Pandorum Technologies, which is focused on corneal blindness, lung and liver diseases, and second, Mestastop Solutions, working on cancer metastasis. He is a member of some of the DBT-BIRAC committees also, including BioNEST and National Biopharma Mission. And with this, I would like to welcome Dr. Ajith Kamath on today's platform to deliver his talk. And also the e-certificates will be given for this webinar to all the participants. For that, you have to mandatorily fill a form by the end of Dr. Ajith's talk that I will share in the chat box. So definitely look up to it. And Dr. Ajith Kamath, over to you.
Oh, thank you. Yeah, thank you very much. If I can share the screen.
Yes, sir, absolutely. Yeah.
You can see my screen?
Yes.
Okay. Is this good?
Yes, sir.
Yeah. Okay. First of all, thank you, Nidhi, for the kind introduction. Thank you, Professor Edana. You have been spearheading, supporting the graduates. I think it's a very noble cause, and all the students and postdocs. I think we are all grateful for your initiatives, and from my point of view, I think the future of India, future of Indian science, drug discovery is all to be run by the youngsters who are all there on this call. And this is an opportunity for me to tell you, how drug discovery is done in India and what are the career opportunities for them. And thanks again, Professor Edana, for giving me this opportunity. Okay. As Nidhi mentioned, I've had a good amount of experience working at Pfizer in the US, and now currently advising a couple of companies. So what I thought was, give you some highlight as to what type of career opportunities are there for life science graduates in India. That would be MSc, MTech, BTech, BSc, PhDs, postdocs, and everybody. So this will be a talk on a theme of how you can contribute to drug discovery and develop some drugs in India. Okay. The topics I'm going to cover, Nidhi told me I have about 45 minutes. So in this 45 minutes, I'll cover the following aspects, give you a brief introduction to the main theme, that is drug discovery. And then we'll briefly talk about various disease areas. And then the most important part for today's talk, that is, talking about career opportunities. To think about career opportunities, you need to think about some skill sets you need and some exposure. We'll discuss that. And then we'll also talk about opportunity to start your own companies. There are fantastic opportunities right now in India with the support of the government, and let's talk about that later part of my talk. Okay. Now, this particular slide, you may have seen it through the Department of Biotechnology. India, has been a leader in many areas, and let's talk about that here. Okay, India has been a destination for vaccines. I'm sure many of you are aware that India is the largest producer of vaccines. About 60% of vaccines that are produced in India is used around the world. Let me put it the other way. 60% of all world supply of vaccines is coming from India. Anybody in the world is taking vaccine, there's 60% chance that that originated from India. I mean, that's a great pride for us in India. Number two is the BT cotton, and we are trying to look at other areas where again India stands huge. Hepatitis B, number 3 producer in the world. That's fantastic news. And then, looking at the second, India has the second highest US FDA approved plants. Okay? If we are anywhere in the world and you are taking a generic tablet for headache or stomach upset or whatever it is, there is about 50% chance that that drug came from India. Okay? That speaks about the capabilities of India in generic field, and that's what is highlighted here. In addition, we have about 3,500 startups. We have significant presence in bioincubators and also share in biotechnology industry, about 3%, which is a great start for a country like India, which has really taken off in the last, I would say, 5 to 10 years in this particular field. Since my talk is on drug discovery, I thought of showing this particular slide to tell you the long road for discovering new drugs. I will not get into the details, but wanted to show you the path. Okay. If we are talking about doing drug discovery, it's a long road. It takes about 10 to 15 years, as you can see at the bottom of this slide. Okay. For various phases of drug discovery, the place where you see a bulb, that is the idea generation, saying that, I want to work on this particular topic. I mean, Loveleen, the sponsor, had discussed about, his particular target. So idea starts from here, saying that we want to come up with a drug for this particular disease. Let's say it's cancer or diabetes, and then a project team is formed that has the biologists, the chemists, the computational chemists, the structural biologists, all of them sit and form a project team. And then you make various compounds. You have to make thousands and thousands of compounds. That's a different discussion altogether, which we can cover some other time. Okay. And then you screen them, identify some nice molecules in the discovery phase. And if it is really a very good nanomolar compound, then it gets into the animal studies. Can you see the cursor moving, Nidhi? No?
Yes, sir.
Okay, yeah. So you get into early animal studies, make sure that the compounds are safe and efficacious, and then you get into a formulation. And that's where the preclinical drug discovery comes to an end. From there, you get into phase 1 in healthy volunteers, and then get into phase 2, carrying out the studies in 100 to 300 patients, and then get into candidate medicine development further in 3,000 to 10,000 patients and up to 30,000 patients. And you may have heard about the vaccines that went to 60,000 patients. So at the end of it, you come up with a molecule that is, you submit to DCGI in India or FDA in the United States, and finally it gets approved. It's a long drawn process, but everything starts from the initial drug discovery phase, which is the topic of today's discussion, and what the opportunities are for all youngsters like you to make a career in drug discovery, and that's what the theme of today's talk. While in the previous slide, I showed you that it takes 15 years to come up with a drug. Look how we addressed the vaccine that we are talking about currently in India. Okay, this is A very, very important slide for all youngsters who are on this call to know as to how, if we put our minds together, if our hearts together, we'll be able to achieve things much faster so that we can help people. Okay? Look at the top of the slide, typhoid fever, vaccine, meningitis vaccine, polio vaccine. They took 20 to 30 years, normally to develop such vaccines. But look at the bottom, SARS-CoV-2 virus vaccine was developed in less than 1 year. Okay, actually it took 248 days for Pfizer from the time they said they want to work on it until they submitted the documents to the FDA. Less than 250 days. This is remarkable because this is a teamwork that does people putting their interest together, solving the problem of the nation, problem of the world, so that they could come up with an innovative vaccine that will touch people's lives. So, there are opportunities. I'm putting up this slide to say that, if we all work together, do some fantastic science, we'll be able to come up with drugs much faster. Okay. So what are the various diseases that are there, that companies in India in particular focus are highlighted here, because you may be thinking about, okay, what research I want to do, what areas I want to work, which are the companies working in various areas. So that's why I put some of that information here, but these are only, the list is exhaustive. I just gave you some examples of diseases that Indian companies are focusing on. It could be on viral diseases. Quite a number of companies are working on cancer, companies working on cardiovascular diseases, metabolic diseases such as diabetes, diseases of the brain, that is CNS diseases, inflammation such as arthritis and other inflammatory diseases, immunology diseases, autoimmune disease, infectious diseases. There are a good number of companies working on infectious diseases. It is something that is very prevalent in India. And of course, there are very few companies working on rare diseases. There's a typo here. Rare diseases, that is also being focused on some of the companies here. Okay. So if you are working in any of these areas, and if you are talking about doing drug discovery, you have to think about novel ways of discovering medicines for those diseases. And that's where my next a few minutes will be talking about. Okay. And as you know, the drug discovery is very, very complex. Okay, we cannot use the same criteria for coming up with a drug what we used 5 years ago, 10 years ago. And you are all the youngsters. You should be thinking about, how do I address the disease much faster? Okay, and come up with a drug that will take 5 years, 7 years, and it should be applicable at those times. So I want you to think about, what will happen in 5 years. Will the drug, will you have a drug, and will the drug be as effective, okay, in 5 years, 10 years? So to support that, lot of development advances has been ongoing in science, and this is one of the examples I wanted to give you about sequencing genome, where right now sequencing genome has become so straightforward. So you'll be able to identify, how do I target one particular disease? How do I come up with a drug for one particular target? And this is just a cartoon to show you, how we can come up with specific enzymes to target a disease, receptors to target a disease, or how do I manage reducing the level of hormones so that the disease can be treated. Okay, so to support that, there are a large number of opportunities for you, all the youngsters on this call. Okay, we have a very different way of looking at disease. Any of these diseases, what we were looking 10 years ago, 15 years ago, okay, when we are coming up with the current drug, the drugs that all of us are taking now, okay, that started 10 years ago, okay, and that time we did not have the understanding that we currently have about the diseases now, okay. The type of targets that we are looking at, probably 10 years ago, people were not able to address those targets. Okay, now is the opportunity for all of you to say how we can, look at some of those targets, protein-protein interactions, and much complex these targets than what was handled in the past. And of course, the technology is also very, very powerful. We can do many things in a very short period of time with high quality data. Okay. I'm just I put up this slide to tease you guys to see how much you can understand about metabolic pathways. I'm not expecting you to read it, but I wanted to show you how massive the metabolic pathway is. And one of these enzymes in the pathways that you see will be the target for the disease. Okay. So to all the people there on the call, I want you to think as to how would you look at one of these enzymes or receptors and say, oh, this particular target is involved in one particular disease, let's say in cancer. Okay, that particular perhaps that enzyme is mutated or it is overexpressed and it is causing cancer. Okay, how do I understand it? And this is what happens in a pharmaceutical industry to do the drug discovery. So if that is the particular kinase that we are looking at, okay, that's what is shown in this particular slide. Nidhi, you can see the slide?
Yes, doctor.
Yeah, okay, yeah. So I'm taking a kinase as an example, and let's assume that this particular kinase is overexpressed, and it is causing, in this particular case, it's a protein kinase, and it phosphorylates the protein from a non-phosphorylated protein. You can see on the left-hand side, it is phosphorylated. You get a phosphorylated protein, and this is the one that is creating the chaos. Okay, a lot more, cells are multiplying and causing the tumor. Okay, so if you want to come up with an inhibitor, you have to come up with an inhibitor for this particular kinase. Okay, let us see how it works in a pharma industry, and let us see, each of you sitting there, tell me how you will think about it to come up with the molecule and where exactly you would fit in. Okay, if this becomes a target in a pharmaceutical company, like what Loveleen was talking about, okay, or Pfizer or Sun and Glenmark and any other companies, Indian companies working on, then they go through this particular process known as phases of drug discovery. Okay, wherein you take the target. That target is the kinase that I showed in the previous slide. You run lots of high throughput screening, identify some molecules called hits, and from there you go to you convert that into a lead. We'll not get into the details, but I'm just trying to set a context for our discussion on job opportunities. Okay, and from there you optimize it and get an optimized lead, and then finally you come up with the candidate which is efficacious and selective and potent. And that's the one that goes into final animal experiments for GLP toxicology studies. Okay, these are the phases of drug discovery, and it is carried out by various scientists, and that is shown in this particular slide. And this is relevant for today's discussion about the opportunities. Okay, so the major components in a drug discovery, I'm talking about small molecules. When I say small molecules, these are organic molecules or natural product derivatives that are made in the lab. And biologics, they are quite different. And I'm focusing primarily on small molecules, tablets or injections and things of that type, organically made. So if you want to do drug discovery, then you have to invariably fall into some of these categories. Among the participants here, there may be chemists here. So chemistry is a huge component in a drug discovery program. People running high throughput screening. There are efforts in some companies to do high throughput screening in India. And of course, as the title of today's presentation is towards life sciences, life science plays a huge role in drug discovery. And that is the biology part. The biochemical part of it comes into the picture, biochemistry, all the enzymology that you learn comes handy here, cell-based assays, carrying out the experiments ex vivo, carrying out in vivo models. You will see some slides later on in my presentation. And then, of course, pharmacology studies are very, very important to know whether the drug is really targeting the right target, target engagement, mechanism of action. All these come under biology. Okay. Once that is done, is the DMPK, drug metabolism, pharmacokinetics part of it, and they are also part of drug discovery. So are the structural biology component of it, trying to understand the structure of protein, doing structure-based drug design, and then of course, computational biology plays an important role in helping out drug discovery. Okay, so if you have had any experience, taken courses, taken projects, written proposals in these areas, then you'll be a good fit for any company working on drug discovery. Okay. I put this particular slide to tell you how do I go about identifying an inhibitor for a kinase that I showed 2-3 slides earlier. Okay, so in this particular case, let's say we make that particular kinase, the protein is a kinase here, and I develop an assay. Okay, in this particular case, phosphorylation assay, but there are many ways of doing it, 10, 15 different ways of carrying out the assay. I carry out the high throughput screening, and then I talk with the medicinal chemists. They make more compounds, and then that is screened again. I use a structure-based drug design effort and selectivity screen to make sure that the molecule that is identified is very unique and it is selective and potent. Once I know that, then I go to the biologist and carry out in vitro studies, in vivo pharmacology studies to make sure that it is doing what it's supposed to do in animal models, and carry out the efficacy study, ADME study, PK/PD studies. And when it is very clean molecule, when I say clean, that means it is targeted, it does not cause toxicity, and it is potent and it's selective, then you say that I have a potent preclinical inhibitor. Okay, so this is what you call it as at the end of it in broad term is a candidate. Okay, so if you want to do this, okay, there are plenty of opportunities, and that's what we'll talk about in the next perhaps 15 20 minutes. That is the opportunity for all of you here on this webinar as to how you can contribute to drug discovery. There are major departments in any drug discovery company or CROs or other companies that are working in the area of drug discovery or providing drug discovery services. That will be a chemistry department, it will be a biology department, it will be a protein science department normally. And then of course, computational biology is part and parcel of many of the companies in India, because it adds immense value in drug discovery program. Okay, so how do you choose a career in a pharma industry? Okay, normally depending upon your interest, depending on the projects that you have carried out in the past and your own motivation, you can look into two different categories. One is called research roles, and the second is called non-research roles in a pharma industry. And we'll get into each of these for the benefit of the audience here. Okay, so let's look into research roles in the pharma, and have categorized them into two. One is function based, and there is a disease based. Okay, these are these are generic classifications. Okay, it could be on there will be a team working on protein purification, protein characterization, the 3D structure. There will be teams working on in vitro based assays, cell-based assays. There will be people working on enzyme kinetics. Many of you would have worked on it. So certainly, that experience of yours, that knowledge of yours comes in handy. If you have studied about mechanism of action, it is something that will be useful here. You would have taken biochemistry classes. It helps molecular biology. If you know what is cloning, what is expression, certainly it will be useful. And so are the others that are given here. In vivo pharmacology, you may have heard about DMPK. You may have taken classes on bioinformatics and metadata analysis, which is very popular nowadays. And of course, anything related to computation modeling. This is on one side. The other side, it would be on disease-based. How do you understand a disease? Okay, what type of signaling pathways are aberrant in this particular disease, causing that particular disease, whether it is inflammation, whether it is cancer, whether it is diabetes. It's all connected with to specific pathways. So how can you control these pathways in order to using drugs and use those drugs for treating patients? How do you validate the target? You may have heard about the knockout models. You may have heard about specific protein inhibitors, or you may have heard of siRNA technology. All of that knowledge comes handy here. And then, of course, the biomarkers are important for disease monitoring, and then translation related, which is a very important area, because if you carry out some studies in animal models, what's the guarantee that they're going to translate in humans? Okay. So are there any specific markers that you can use them to show that indeed these markers are are there in animals, and the drug regulates the level of these biomarkers, and similar thing can be done in humans. So the translation comes into the picture too. Okay. Since there may be chemists in this audience, I put this particular slide to tell you various aspects of chemistry that goes on in a CRO, in a drug discovery. That will be medicinal chemistry, making compounds, analytical chemistry, making sure that the compound that are prepared are characterized by HPLC, mass spec, NMR, crystallography, X-ray crystallography. Then, process chemistry, the molecules are initially made in milligram quantities, perhaps 2 milligrams, 5 milligrams. And when project really works, then you have to make 10 milligrams to, 10 grams. And if not, eventually to make, a kilogram quantities. That's the scale-up lab. You have to have library design, and green chemistry is something that I'm sure many of you have heard about it, and it's very critical in industry because you want to, save the environment, create less pollution. So that is, I gave one particular slide on chemistry. Let's look at computational biology for students sitting in this meeting. Okay, it's a very, very important component of drug discovery, looking at the sequence and then predicting their function, or looking at the homology and saying, okay, this is what the function is, doing homology modeling, coming up with the structures. And then, of course, once you have the structure, you can dock the potential compounds. You can do screening of molecules by in silico screening and come up with the structure and where the possible docking, the molecule gets docked. Okay, docking of the ligands, and these are all very critical components of drug discovery, and there is significant effort in India to support computational biology. And I hope some of you have taken computational biology courses, and that will help. Okay. The other area would be the bioinformatics. Many of you would have taken courses in bioinformatics, and I'm using this slide to tell you the power of bioinformatics in drug discovery. Start trying to understand molecular interaction of molecules. It could be biochemical networks, or it could be analyzing sequences and converting that into protein, trying to understand their function, and then doing a lot of variations, genetic variations. Why would Indian population with Indian genotype would differ from what happens in the Western world? Their genome is different. Would Indian patients response similarly to the drug that was developed in other countries. These are all important, and bioinformatics provides some of those tools to understand how we can maximize on the genome information and ensure that the drugs that we develop are utilized maximally with the least side effects. Okay. I will, in the interest of time, I'll skip this slide. What happens in protein science? I wanted to give you an example. If many of you are, masters and you are not, doing MSc, MTech, and you are not seeing what happens. I'll take you through protein science as an example with regard to, how work is done in the lab. Okay. Let's say a project comes to the lab. I hope you have done some practicals in the lab. Okay, so you start saying, remember the kinase that we talked about? We want to make a synthetic gene, put it in one particular vector, either E. coli or insect cells or mammalian cells, and transform or transfect that particular cell line, and then you grow the cells. Okay, once you grow the cells, you centrifuge, you purify the protein through affinity chromatography, FPLC, gel filtration chromatography, and then use that for all the assays. This is what is done in a protein science team. So if you have had some experience and somebody says, okay, you are joining the protein science team, this is what you'll be doing. Either you will get into a lot of molecular biology work of cloning, or you will help in protein purification that you can see here, protein characterization as shown here. Okay, look at the peak here, shows you the purity of the protein characterization, that you did a very good job in purifying high quality protein. And this particular protein is crystallized, and this is the structure of the protein with the inhibitor bound. And this is the active site of the protein, which is solved by X-ray crystallographer, and a computational biologist also helps. Okay, so in protein science team, if you join such a team, then you'll be part and parcel of each of these, doing somebody within the team growing cells, somebody doing the cloning part, somebody doing pure protein purification, and somebody else solving the structure and saying, hey, as a team, we solved the structure of this particular protein. All we started was with the clone. Now we have the purified protein, we have the structure, and we know where the compound binds. Okay. There could be other roles within protein science is cell culture. Many of you may have grown small quantities of cells as a part of your project or your research, 5 ml, 10 ml, E. coli culture or mammalian culture, and that's what happens in a pharma industry too. Okay, you create small cultures, you do some initial cloning cell culture and grow it in small scale, freeze it, or you make various cell lines as is shown here in the bottom half of the slide. But then you really want to scale up, you go to bigger companies. Okay, and this is how the biologics are purified. I'm talking about biosimilars, which have to be grown in 100, 200, 300, 1,000 liters, 10,000 liter fermenters, and you see some of these fermenters here. Somebody is working there, growing the cells. I took this from a Jubilant Biotech company based in Chennai from their website. This is very illustrious as to how this can be done. On the right-hand side, you see somebody purifying the protein. If many of you in the audience have purified protein in 1 ml column, 2 ml column, I want you to pay attention to the one right here in the middle that I'm showing. This is preparative, large scale. This must be a 5 liter, maybe 4 liter, 5 liter column that you use to purify, and you will get, instead of milligram proteins, you are getting kilogram proteins. And this is the protein. It could be insulin, it could be growth factors, it could be antibodies for rheumatoid arthritis or macular degeneration or any disease. Okay, so this is again, employing your knowledge, how you do it. It's not that they'll take you and say, okay, from tomorrow onwards you start operating this machine. That's not what happens. They'll train you very well, but you have to know the basics. And this is the protein that eventually will go to the patients. Okay. If you are into animals, I showed one particular slide here. If you are in the regular in vivo pharmacology study, then you have to, this is the mouse, you create the tumor. This is a cancer project. You create the tumor in the mouse, and then you treat this particular mouse with the drug that we talked about, the kinase inhibitor. Let's say that is a very powerful, very potent drug. You give that kinase inhibitor to this particular mouse and see whether the size decreases. Okay. Then you have to monitor, look at the size of the tumor, look at the histology of the tumor, carry out quite a number of studies. This again should give you some idea as to, what are all the various areas of research that goes on in the pharmaceutical industry. Okay. So far I talked about roles within what we call it as a wet lab. Okay, that means you sit there, you run gels, you run chromatography, you grow cells, you take care of animals, you make compounds, you run projects, and make sure that it's all wet lab work. The other roles within pharma is given in this slide. Okay, this talks about the intellectual property. People with masters, if they can get into intellectual property, carry out some, IPR courses, that is excellent combination, because you understand the biology or the chemistry in life science, and now you follow the rules of patent filing. What is required for patents? How do you understand it? If it's a regular IP lawyer, intellectual property lawyer, he or she may not understand the biology. Whereas if you have a biology background, then you fit in very well, because you understand what a clone means, what a sequence means, why a particular mutation is the one that's causing that disease. You understand, but not the legal company, right? So from that point of view, intellectual property is a very good area to be in. There are openings in project management. People can do the work, but everything has to be documented, everything has to be monitored very, very closely. Each project has their own timeline. So that is taken care of by the management, project management. Okay. Regulatory aspects. Remember how I told you what goes on in the lab, and then it has to be scaled up, and it has to be done in a GMP facility where the rules are very, very strict. Everything is very much followed, because that is the material that goes into the patients, and that documentation has to be done, and it is submitted to the DCGI. So that is the regulatory aspects. Okay. And then of course, data science is there, clinical data analysis, quality control to make sure that the data that's generated is of the highest quality and there is consistency from batch to batch. Quality is that most important part. You can't say, last time I got a very good band and this time I'm not getting it. That can't happen. It has to be identical to the result that you got previously, and that's all monitored by the quality control department. And of course, environmental health and safety, they're all there. I put this up because there may be people on this call from environmental sciences, people, from ocean, marine biology background. So I put some of these, for their benefit. So in addition to the lab work, which we discussed all along, this is one slide I incorporated to show you what other roles are available within pharma or biotech or non-wet lab work. Okay. I have a few more slides to talk about what is the requirement. Okay, all of you are youngsters, all of you are studying very, very hard. So I want you to spend a little bit more time thinking about what you want to do. Okay, if you want to join an industry, then you have to have a very good understanding about the fundamentals of all aspects of your coursework or the project. Okay. It is very important. It's nobody in the industry expects you to come and start working the next day on a very difficult project. They want to know how sound is your understanding of the courses that you have taken. okay? How well you understood, how you did the troubleshooting, how did you come up with the answer, okay? That is what they're looking for. And if you have carried out any projects, how did you, why did you decide on that project? How did that project, you know, how did you address the problem, address the questions, and what was your conclusion? So all these things requires quite a bit of dedication, and that is what is expected in if you are a master's or PhD student or a postdoc, okay? And then try to understand what are the areas that you are very, very confident in, okay? I'm talking about the science that you're doing with regard to your work, okay? If you say that I am very good in this particular field, I fully understand, I mean, let me just take an example, okay? That you are very much into, metabolism of in human beings, okay? Then that is your strength. You may not know as much as perhaps in the immunology area, but you may be very good in metabolism, okay? So some of those areas where you want to pursue your career in, you have to know this is where my strength is and this is what I want to pursue, okay? That is the next bullet I had, specific areas that you want to pursue. I think it's very important for you to know what you want to do, but you also want to know what areas you don't want to work on, okay? When I was studying, I was mostly into biochemistry. I wanted to do pathways. I wanted to identify inhibitors. I wanted to do a lot of protein characterization, protein properties, mutations, effect of mutations, mechanism of action, and all these things. I was not too much into carrying out studies in animals. Okay, so my preference was mostly on the biochemistry side, enzymology side, kinetic side, rather than in vivo pharmacology side. Okay, I understand pharmacology side, but that's not the area where I was trained. Whereas I was trained as a biochemist. So it is good for each of you to know what your strengths are and where you want to pursue your career in. Okay, so if you have those things in mind, then how would it match to an industry? Okay, if you want to do a career in industry, okay, please think about incorporating some of that in your regular work that you're doing, whether you're doing your coursework, whether you're doing your PhD or your postdoc. Please consider some of these things that are captured here, okay? Carry out a research project. If there is an opportunity for you to do it, please do it, okay? And or write a thesis, okay? Because that gives you a better understanding of why you are working on that particular project and how it helped. Okay? If you are working in that particular area, then your thought process is very, very good. Okay, write a project proposal. I'm sure you have had some projects where in your masters or BTech or BSc or PhD, they'll say, okay, now write this particular proposal. Please take initiative to do it because it helps you to get a better understanding of that particular project, whether it is drug discovery or anything, plants, it doesn't matter, okay? And try to get some hands-on experience either in companies, okay, or in research labs. Whenever you have an opportunity, please use it and work somewhere as an intern or whichever way. Reach out to your supervisors and ask them, can you please refer me to somebody? Can you please connect me to somebody? You have to take that initiative, and that helps you a lot to get to know what happens in the industry, and you'll be better prepared when you join the industry. Exposure to new technologies, okay, either through your coursework when you're studying, please pay attention to new technologies that are coming up, or read journals, read scientific articles and see how things have changed, because that is the future, and that is your future, that is our future. Okay, and awareness of new developments in the area of interest. Okay, if you are interested in diabetes, we cannot use the same medicine that we used 20-30 years that were developed 20-30 years ago. Now diabetes treatment is very different than what it was done before. So any area that's of interest, please look into that. It might be on plants, getting a better plant, okay, or marine biology. I'm just bringing drug discovery as an example. And then try to do literature analysis and improve your presentation skills, give presentations in your seminars in the department, or talk to your friends, give presentations. These are all important skills that the industries will look for when you are interviewing in those places. Okay. I just put the pharma companies, here, the top companies in India. Many of them are the leaders in generics and also in some of the drug discovery efforts. Okay. All right. So in next 5 minutes or 10 minutes, Nidhi, let me talk about opportunities in biotech startups. Okay. All my previous slides focused on how you can get into a drug discovery company, perhaps like Cipla, Cadila, Glenmark, Sun, Intas, Biocon, and Reddy's, and everybody else, Panacea, any field that you want to work on. Or you can think about various CROs like Syngene, Jubilant, Origin, GVK, and many other companies, including the one that Lovelin talked about. And thanks, Lovelin, for sponsoring. Okay. Now look at in a different way. How about some of you? I think we have a good amount of people joining the call. You want to start your own. Are there any opportunities in India for doing that? Okay, and the answer is certainly yes. Okay, look at this slide. Okay. India's economy, bioeconomy, is $70 billion and is expected to double in next 5 years. There are 3,500 startups, and the aim is to go to 10,000 in next 5 years. Okay. And there are 350 plus incubators, large number of them into the biotech area. So some of you, if you want to take a challenge saying that, I want to start something on my own, how do I do that? Who is there to help me? Okay. What type of encouragement is there by the government, and what is going on in India? Okay. The answer is, looks very promising. In the last 5 to 7 years, thanks to Department of Biotechnology and BIRAC. And you may have just seen this announcement by the Prime Minister last month, okay, announcing 1,000 crore Startup India seed fund, okay. And as you can read here, okay, there will be 3,600 healthcare companies in startups, and 1,700 startups in agriculture. These are huge numbers, and it is a fantastic opportunity for any of you who are looking into this area. And I put an arrow to his comments where he says, we are trying to create a startup system which is based on the mantra of the youth, by the youth, and for the youth. And I'm sure all the people listening to in this webinar, they all fall into this category. Okay, you are the future of this country, and that's what the Prime Minister is talking about, that you have to take the initiative to set up your own company, set up your own startup, and see how you can come up with products that will benefit people in India and across the world. Okay, one effort that goes on is BIRAC. As Nidhi said, I participate in many of the meetings with BIRAC on many of their committees, and BIRAC helps in agriculture, healthcare, clean energy, industrial, biotech and medtech, and then of course biopharma is all a part of the objectives for BIRAC to support. If you have some ideas, this slide talks about a lab to market and what does BIRAC do. Okay, the slide at the bottom shows students. I'm sure many of you are students, you are researchers. If you have that entrepreneurial spirit in your heart, I think you have to jump into this and say how I can benefit from the schemes of the Government of India. You can say, I don't know much, I have some cool ideas that I want to work and is going to help, the society. There are multiple ways you can engage, BIRAC to go from your idea stage to proof of concept stage, early development, late development, and finally to the market. Okay. To do this, you need a lot of support. You don't have to do everything by yourself. You know, the BIRAC will help you with the initial funding. They'll help you capacity building. They'll provide you some space. They'll provide you mentorship. Okay, and various things that are shown here on the left-hand side. Okay, that is the kind of opportunity you youngsters have compared to what was missing perhaps 10 years ago, 15 years ago, 20 years ago. This is unheard of. So I encourage you to think about how you can take your own ideas into the lab, come up with some initial proof of concept studies, and then try to see whether you can take it to the market. Okay. To do all these things, you require a lot of money, and who gives you the money? And that is shown here in this particular slide. Okay. This is all the funding that the government gives. This is BIRAC funding. They have quite a number of schemes to help youngsters to make their career in startups. Okay, they have various schemes. These are various schemes. Sitare, BIG, SIBRI grant, BIPP grant, Seed grant, LEAP grant. Okay, quite a number of grants that the government gives. And they give the space in a place called BioNEST, University of Hyderabad, where Professor Reddanna is. That is a BioNEST incubator. They have a BioNEST incubator. So there are, a lot of opportunities for all you youngsters to start up your own, if that is something that you're thinking about. Okay. My last slide is some of the products that BIRAC supported with people like you who are participating in this webinar, funded by the government, and these products are currently available for the needy in India. Okay, and then this is I think this is the last slide. Make in India and Startup India. I got this slide from BIRAC. They were kind enough to give me some slides that I could use, and just to encourage some of you to look into starting your own company. Okay. So the key takeaways, going back to the theme of my presentation, whether you are looking for a job in a drug discovery company, whether you're looking for a job in a CRO, or you want to start your own, okay, these are the takeaways. Okay, identify the area or areas that you would like to work on, whichever it might be, marine biology or environmental biology, or chemistry, life science, anything. Just identify one particular area and pursue that. Okay, but you have to learn more in that particular field with examples. And say, this is what I want to do. This is how the current products are, but I want to get far better drugs or molecules than what it is. Okay, or I want to develop better technology for diagnosis than what it is now. Okay, so each of them will help you to refine your thought process and take it forward. Explore working in that area in an academic lab. I mentioned this before. If there is an opportunity to go and work in the lab, get some hands-on experience, that will help. Explore internship in companies. Reach out to your professors, do some networking, and say, sir, please help me find an internship somewhere. And if it gives you a better perspective as to what happens in the industry. And then show genuine interest and enthusiasm in your coursework and the project that you're working on. Okay, don't put any half-hearted attempts. Put your heart and soul into this. Show dedication. Show a lot of interest. Take this project forward, and that's it. That's what will help you in your projects, in your degree. But same thing will help you when you go for an interview, or when you take up a job in the industry. Okay? Whether it's your own company or any other's company or drug discovery company. And the last one is read about recent developments. Things are changing very, very fast. I just gave an example as to how quickly people were able to come up with the vaccine, which took decades. Now it is taking less than a year. Same thing with regard to sequencing. It used to take years. Now it is taking perhaps a week. Okay. So a lot of developments have happened. So be on the top of it. That will help you in your career. With that, I'll stop it here. I think that's the last slide. Nidhi, again, I specifically want to thank Professor Redana for taking this initiative. Professor Redana, Nidhi, you did a fantastic job. And then, Loveleen, thank you very much for sponsoring. Back to you, Nidhi.
Thank you so much,